Hello everyone, my name is Sneha. I am a student of BSc Chemistry Honours 6th semester. Today I will make a video for visually impaired students on the topic organic mechanisms of the coenzymes metabolism. Many enzymes cannot catalyze a reaction without the help of a cofactor. Cofactors assist the enzymes in catalyzing a variety of reactions that cannot be catalyzed solely by the amino acid side chains of the protein. Some cofactors are metal ions while others are organic molecules. A metal ion cofactor acts as a Lewis acid in a variety of ways to help an enzyme catalyze a reaction. It can coordinate with groups on the enzyme, causing them to align in a geometry advantageous for reaction. It can help bind the substrate to the active site of the enzyme. It can target for reaction. It can form a coordination complex with the substrate to increase its reactivity, or it can increase the nucleophilicity of water at the active site. An enzyme that has a tightly bound metal ion is called metalloenzyme. Carboxypeptidase A is an example of metalloenzyme. Cofactors that are organic molecules are called coenzymes. Coenzymes are derived from organic compounds commonly known as vitamins. Table 25.1 lists the vitamins and their biochemically active coenzyme forms. A vitamin is a substance that body cannot synthesize that is needed in small amounts for normal body function. Sir Frederick Hopkins was the first to suggest that the diseases such as rickets and scurvy might result from the absence of substances in the diet that are needed only in very small quantities, because the first such compound recognized to be essential in the diet was an amine. Casimir Funk incorrectly concluded that all such compounds were amines and called them. Now, coenzymes play a variety of chemical roles that the amino acid side chain of enzymes cannot play. Some coenzymes function as oxidizing and reducing agents, some allow electrons to be delocalized, some activate groups for further reaction, and some provide good nucleophiles and strong bases needed for a reaction, because it would be highly inefficient for the body to use a compound only once and then discard it. Coenzymes are recycled. Therefore, we will see that any coenzyme that is changed during the course of a reaction is subsequently converted back to its original form. An enzyme plus the cofactor it requires to catalyze a reaction is called a haloenzyme. An enzyme that has its cofactor removed is called an apoenzyme. Holoenzymes are catalytically active, whereas apoenzymes are catalytically inactive because they have lost their cofactors. Early nutritional studies divided vitamins into two classes: water-soluble vitamins and water-insoluble vitamins. Vitamin A, D, E, and K are water-insoluble. Vitamin K is the only water-insoluble. Early nutritional studies Vitamin K is the only water-insoluble vitamin currently known to function as a coenzyme. Vitamin A is required for proper vision, and vitamin D regulates calcium and phosphate metabolism, and vitamin E is an antioxidant because they do not function as coenzymes. Vitamin A, D, and E are not discussed in this chapter. Vitamin A and E are discussed in section 9.A and 26.7, and vitamin D is discussed in the next section. All the water-soluble vitamins except vitamin C functions as coenzymes. In spite of its name, vitamin C is not actually a vitamin because it is required in fairly high amounts. And most mammals are able to synthesize it. Humans and guinea pigs cannot synthesize it, however, so it must be included in their diets. We have seen that vitamin C and vitamin E are radical inhibitors and therefore are antioxidants. Vitamin C traps radicals formed in aqueous environments, whereas vitamin E traps radicals formed in nonpolar environments. It is impossible to overdose on water-soluble vitamins because the body can readily eliminate any excess. You can, however, overdose on water-insoluble vitamins because they are not easily eliminated by the body and can accumulate in cell membranes and other non-polar components of the body. For example, excess vitamin D causes calcification of soft tissues. The kidneys are particularly susceptible to calcification, which eventually leads to kidney failure. Vitamin D is formed in the skin as a result of photochemical reaction. The reactions that living organisms carry out to obtain the energy they need and to synthesize the compounds they require are collectively known as metabolism. The process of metabolism can be divided into two parts, catabolism and anabolism. Catabolic reactions break down complex nutrient molecules to provide energy and simple precursor molecules for synthesis. Anaerobic reactions require energy and result in the synthesis of complex biomolecules from simpler precursor molecules. Catabolism, complex molecules give simple molecules plus energy. Anabolism, simple molecules plus energy gives complex molecules. It is important to remember that almost every reaction that occurs in a living system is catalyzed by an enzyme. The enzyme holds the reactants and any necessary cofactors in place so that the reacting functional groups and catalytic groups are oriented in a way that will cause a specific well-defined chemical reaction. In some cases, it may be helpful to see where a particular enzyme catalyzed reaction discussed in this chapter fits into the overall metabolic scheme. Catabolism can be divided into four stages. The first stage of catabolism is called digestion. The reactants required for all the life processes ultimately come from our diet. In that sense, we really are what we eat. In this first stage, fats, carbohydrates, and proteins are hydrolyzed into fatty acids, monosaccharides, and amino acids, respectively. In the second stage of catabolism, these hydrolysis products, fatty acids, monosaccharides, and amino acids, are converted to compounds that can enter the citric acid cycle. To enter the citric acid cycle, a compound must be either one of compounds in the cycle itself called a nitric acid cycle intermediate, acetyl cofactor, coenzyme. Acetyl coenzyme is the only non-citric acid cycle intermediate that can enter the cycle. Pyruvate can enter the citric acid cycle only because it can be converted to acetyl coenzyme A or to oxaloacetate, a citric acid cycle intermediate. Fatty acids are converted to acetyl coenzyme, monosaccharides are converted to pyruvate via glycolysis, and amino acids are converted to acetyl coenzyme A, pyruvate, and citric acid cycle intermediates depending on the amino acid. The third stage of catabolism is the citric acid cycle, Krebs cycle, or tricarboxylic TCA cycle. For every acetyl coenzyme A that enters the cycle, 2 molecules of CO2 are formed. Metabolic energy is measured in terms of adenosine triphosphate ATP. How the body uses ATP is described in section 27.2 and 27.3. Very little ATP is formed in the first 3 stages of catabolism. However, in the fourth stage of catabolism, every NADH that is formed in the process of carrying out oxidation reactions in the earlier stages is converted into 3 ATPs in a process known as oxidative phosphorylation. Thus, the bulk of the energy provided by fats, carbohydrates, and proteins. Anabolism can be thought of as the reverse of catabolism. In anabolism, acetyl coenzyme A, pyruvate, and citric acid cycle intermediates are the starting materials for the synthesis of fatty acids, monosaccharides, and amino acids. These compounds are then used to form fats, carbohydrates, and proteins. The mechanisms utilized by biological systems to synthesize fats and proteins are discussed in section 19.21 and 27.13. Here, glucose changes into glucose 6-phosphate in the presence of magnesium 2 positive ion and hexokinase Then this glucose 6 phosphate changes further. Lysine, the vitamin needed for many redox reactions. An enzyme that catalyzes an oxidation reaction or a reduction reaction requires a coenzyme because none of the amino acid side chains can act as oxidizing or reducing agents. The coenzyme is the oxidizing or reducing agent. The enzyme's role is to hold the substrate and coenzyme together so that the oxidation or reduction reaction can take place. The coenzymes most commonly used for enzymes to catalyze oxidation reactions are nicotinamide adenine dinucleotide NAD positive and nicotinamide adenine dinucleotide phosphate NADP plus. When NAD plus or NADP plus oxidizes a substrate, the coenzyme is reduced to NADH or NADPH. NADH and NADPH are reducing agents. They are used as coenzymes by enzymes that catalyze reduction reactions. Enzymes that catalyze oxidation reactions bind NAD plus or NADP plus more tightly than they bind NADH or NADPH. When the oxidation reaction is over, the relatively loosely bound NADH or NADPH dissociates from the enzyme. Likewise, enzymes that catalyze reduction reactions bind NADH more tightly than they bind NAD plus or NADP plus. When the reduction reaction is over, the relatively loosely bound NAD plus or NADP plus dissociates from the enzyme. NAD plus is composed of two nucleotides linked together through their phosphate groups. A nucleotide consists of heterocycle attached in a beta configuration to C1 of a phosphorylated ribose. A heterocycle is a cyclic compound in which one or more of the ring atom is an atom. The heterocyclic component of one of the nucleotides of NAD plus is nicotinamide and the heterocyclic component of the other is adenine. This accounts for the coenzyme's name nicotinamide adenine dinucleotide. The positive charge in the NAD plus abbreviation indicates the positively charged nitrogen of the substituted pyridine ring. The only way in which NADP plus differs structurally from NAD plus is in the phosphate group bonded to the 2-OH group of the ribose of the adenine nucleotide. This explains the addition of phosphate to the name. NAD plus and NADH are generally used as coenzymes in catabolic reactions. NADP plus and NADPH are generally used as coenzymes in anabolic reaction. The adenine nucleotide for the coenzymes is provided by ATP. The oxidation of the secondary alcohol group of malate to a ketone group is one of the reactions in the citric acid cycle. NAD plus is the oxidizing reagent in this reaction. Many enzymes that catalyze oxidation reactions are called dehydrogenases. Recall that the number of CH bonds decreases in an oxidation reaction. In other words, dehydrogenases remove hydrogen. Beta aspartate semialdehyde is reduced to homoserine in an anabolic pathway. The differentiation between the coenzyme used in catabolism and anabolism is maintained because the enzymes that catalyze these oxidation reduction reactions exhibit strong specificity for a particular coenzyme. For example, an enzyme that catalyzes an oxidation reaction can readily tell the difference between NAD plus and NADP plus. If the enzyme is in a catabolic pathway, it will bind NAD plus but not NADP plus. In addition, the relative concentration of the coenzymes in the cell encourage binding of the appropriate coenzyme. For example, because NAD plus and NADH are catabolic enzymes and catabolic reactions are most often oxidation reactions, the NAD plus concentration in the cell is much greater than the NADH concentration. Because NADP plus and NADPH are anabolic coenzymes and anabolic pathways are predominantly reduction reactions. The concentration of NADPH in the cell is greater than the concentration of NADP+. The ratio of NADP plus upon NADPH is maintained about 0.01. Mechanisms for pyridine nucleotide coenzymes. How do these oxidation reduction reactions take place? All the chemistry of the pyridine nucleotide coenzymes NADP plus, NAD plus, NADH and NADPH takes place at the fourth position of the pyridine ring. The rest of the molecule is present for binding the coenzyme to the proper site on the enzyme. If a substrate is being oxidized, it donates a hydride ion H negative to the fourth position of the pyridine ring. In the following reaction, the primary alcohol is oxidized to an aldehyde. A basic amino acid, glyceraldehyde 3-phosphate dehydrogenase is an example of an enzyme that uses NAD plus as an oxidizing coenzyme. The enzyme catalyzes the oxidation of the aldehyde group of glyceraldehyde 3-phosphate to an anhydride of a carboxylic acid and phosphoric acid. This is a reaction that occurs in glycolysis. In the first step of the mechanism for this reaction, an SH group of a cysteine side chain at the active site of the enzyme reacts with glyceraldehyde 3-phosphate to form a tetrahedral intermediate. A side chain of the enzyme increases cysteine's nucleophilicity by acting as a general base catalyst. The tetrahedral intermediate expels a hydride ion, transferring it to fourth position of the pyridine ring of an NAD plus that is bonded to the enzyme at an adjacent site. NADH dissociates from the enzyme and the enzyme binding a new NAD plus phosphate reacts with the thioester, forming the anhydride product and releasing cysteine. Notice that at the end of the reaction, the hollow enzyme is exactly as it was at the beginning, so the catalytic cycle can be repeated. The NADH produced in the reaction is reoxidized to NAD plus in the fourth stage of catabolism. The mechanism for reduction by NADH or by NADPH is the reverse of mechanism for oxidation by NAD plus or by NADP plus. If a substrate is being reduced, the dihydropyridine ring donates a hydride ion from its fourth position to the substrate. An acidic side chain of the enzyme aids the reaction by donating a proton to the substrate. Because NADH and NADPH reduces compounds by donating a hydride ion, they can be considered biological equivalents of NaBH4 and LiAlH4. Hydride donors used as reducing reagents in non-biological reactions. Why are the structures of biological redox, reducing and oxidizing reagents so much more complicated than the structure of the redox reagents used to carry out the same reactions in the laboratory? NADH is certainly more complicated than LiAlH4, although both reagents reduce compounds. Molecular recognition allows the enzyme to bind the substrate and the coenzyme in the proper orientation for reaction. In addition, a redox reagent found in a biological system must be less reactive than a laboratory redox reagent because it must be selective. For example, a biological reducing agent cannot reduce just any reducible compound with which it comes into contact. Biological reactions are much more carefully controlled than that because the coenzymes are relatively unreactive compared with non-biological redox reagents. The reaction between the substrate and the coenzyme does not occur at all or take place very slowly without the enzyme. For example, NADH will reduce an aldehyde or a ketone only in the presence of an enzyme. NaBH4 and LiAlH4 are more reactive hydride donors, much too reactive to exist in the aqueous environment of the cell. Similarly, NAD plus is a much more selective oxidizing agent than the typical oxidizing agents used in the laboratory. For example, NAD plus will oxidize an alcohol only in the presence of an enzyme. Because a biological reducing agent must be recycled rather than having its oxidized form thrown away as would be the case for a reducing agent used in the laboratory, the equilibrium constant between the oxidized and reduced forms is generally close to unity. Therefore, biological redox reactions are not highly exergonic, rather, they are equilibrium reactions that are driven in the appropriate direction by the removal of reaction products as a result of participation in subsequent reactions. As you study the coenzyme in this chapter, don't let the complexity of their structures deter you. Notice that only a small part of the coenzyme is actually involved in the chemical reaction. As Notice also that the coenzymes follow the same rules of organic chemistry as do the simple organic molecules with which you are familiar. We saw in section 5.16 that an oxidizing enzyme can distinguish between two hydrogens on the carbon. Similarly, a reducing enzyme can distinguish between two hydrogens at the fourth position of the nicotinamide ring of NADH. An enzyme has a specific binding site for the coenzyme, and when it binds the coenzyme, it blocks one of its sites. If the enzyme blocks the B side of NADH, the substrate will bind to the A side and the HA hydride ion will be transferred to the substrate. If the enzyme blocks the A side of the coenzyme, the substrate will have to bind to the B side and HB hydride ion will be transferred. Currently, 156 dehydrogenases are known to transfer HA and 121 are known to transfer HB. Flavin adenine dinucleotide and flavin mononucleotide vitamin B2. A flavoprotein is an enzyme that contains either flavin adenine dinucleotide FAD or flavin mononucleotide FMN as a coenzyme. FAD and FMN, like NAD plus and NADP, are coenzymes used in oxidation reactions. As its name indicates, FAD is a dinucleotide in which one of the heterocyclic components is flavin and the other is adenine. FMN contains flavin but not adenine. It is a mononucleotide. Flavin is a bright yellow compound. Flavus is Latin for yellow. Notice that instead of ribose, the flavin nucleotide has a ribitol group, a reduced ribose. How can we tell which enzyme use FAD or FMN rather than NAD plus or NADP plus as the oxidizing coenzyme? A rough guideline is that NAD plus and NADP plus are the coenzymes used in the enzyme catalyzed oxidation reactions involving carbonyl compounds, alcohols being oxidized to ketones, aldehydes or carboxylic acids, while FAD and FMN are the coenzymes used in the other types of reactions. For example, in the following reactions, FAD oxidizes a dithiol to a disulfide, an amine to an imine, and a saturated alkyl group to an unsaturated alkene. Then FMN oxidizes NADH to NAD+. This is only an approximate guideline. However, because FAD is involved in some oxidations that involve carbonyl compounds, and NAD plus and NADP plus are involved in some oxidations that do not involve carbonyl compounds. Mechanisms for flavin nucleotide coenzymes. When FAD or FMN oxidizes a substrate S, the coenzyme is reduced to FADH2 or FMNH2. FADH2 and FMNH2, like NADH and NADPH, are reducing agents. All the oxidation reduction chemistry takes place on the flavin ring. Reduction of the flavin ring disrupts the conjugated system, so the reduced coenzymes are less colored than their oxidized forms. In the first step of the mechanism, for the FAD catalyzed oxidation of dihydrolipoate to lipoate, the thiolate ion attacks the C4A position of the flavin ring. This reaction is general acid catalyzed. As the thiolate ion attacks the ring, a proton is donated to the N5 nitrogen. A second nucleophilic attack by a thiolate ion, this time on the sulfur that is covalently attached to the coenzyme, generates the oxidized product and FADH2. Section 25.4 discusses where this FAD catalyzed reaction fits into metabolism. In the first step of the flavin catalyzed oxidation of an amino acid to an amino acid, a basic side chain at the active site of the enzyme removes a proton from the alpha carbon of the amino acid. The carbon ion that is formed attacks the N5 position of the flavin ring Collapse of the resulting tetrahedral intermediate gives the oxidized amino acid. Notice the FAD is a more versatile coenzyme than NAD+. Unlike NAD+, which always uses the same mechanism, flavin coenzymes can use several different mechanisms to carry out an oxidation. For example, we have just seen that when FAD oxidizes dihydrolipoate, nucleophilic attack occurs on the C4A position of the flavin ring, but when it oxidizes an amino acid, nucleophilic attack occurs on the N5 position. Cells contain very low concentrations of FAD and much higher concentrations of NAD+. This difference in concentration is responsible for a significant difference in the enzyme E that use NAD+ as an oxidizing agent and those that use FAD. Generally, FAD is covalently bound to its enzyme and remains bound after being reduced to FADH2. Unlike NADH, FADH2 does not dissociate from the enzyme. It therefore must be reoxidized to FAD before the enzyme can enter another round of catalysis. The oxidizing agent used for this reaction is NAD+ or O2. Therefore, an enzyme that uses an oxidizing coenzyme other than NAD+ may still require NAD+ to reoxidize the reduced coenzyme. For this reason, NAD+ has been called the common currency of biological. Thiamine pyrophosphate, vitamin B1. Thiamine was the first of the B vitamins to be identified, so it became known as vitamin B1. The absence of thiamine in the diet causes a disease called beriberi, which damages the heart and impairs nerve reflexes. In extreme cases, it causes paralysis. One major dietary source of vitamin B1 is the hulls of rice kernels. A deficiency is therefore most likely to occur when highly polished rice is a major component of the diet. A deficiency is also seen in alkalis. The coenzyme form of vitamin B1 is thiamine pyrophosphate (TPP). TPP is the coenzyme required by enzymes that catalyze the transfer of a 2-carbon fragment from one species to another. Pyruvate decarboxylase is an enzyme that requires thiamine pyrophosphate. Pyruvate decarboxylate catalyzes the decarboxylation of pyruvate and transfers the resulting 2-carbon fragment to a proton, resulting in the formation of an alpha keto acid. You may wonder why an alpha keto acid such as pyruvate can be decarboxylated, since the electrons left behind when CO2 is removed cannot be delocalized onto the carbonyl oxygen. We will see that the thiazolium ring of the coenzyme provides a site for the delocalization of the electrons. A site to which electrons can be delocalized is called an electron sink. Mechanism for pyruvate decarboxylase. The hydrogen bonded to the imine carbon of TPP is relatively acidic because the lead carbanion formed when the proton is removed is stabilized by the adjacent positively charged nitrogen. The lead carbanion is a good example of this. In the first step of mechanism for pyruvate decarboxylase, the nucleophilic carbanion attacks the electrophilic ketone group of alpha keto acid. The resulting intermediate can easily undergo decarboxylation because the electrons left behind when CO2 is removed can be delocalized on the positively charged nitrogen. The positively charged nitrogen of TPP is a more effective electron sink than the beta keto group of a beta keto acid, a class of compounds that are fairly easily decarboxylated. The product of decarboxylation is stabilized by electron delocalization. One of the resonance contributions is neutral and the other has separate changes. We will call it a resonance stabilized carbanion. Protonation of the resonance stabilized carbanion and a subsequent elimination reaction form acetaldehyde and regenerate the coenzyme capable of entering the citric acid cycle. For the final product of carbohydrate metabolism is pyruvate. To enter the carboxylic acid cycle, pyruvate must be converted either to acetyl coenzyme A or to oxaloacetate, a citric acid cycle intermediate. The pyruvate dehydrogenase system is a group of 3 enzymes responsible for the conversion of pyruvate 2-acetyl coenzyme. A. The pyruvate dehydrogenase system requires TPP and 4 other coenzymes. The first enzyme in the system catalyzes the reaction of TPP with pyruvate to form the same resonance stabilized carbon ion formed by pyruvate decarboxylase and by the enzyme in problems 8 and 9. The second enzyme of the system E2 requires lipoate, a coenzyme that becomes attached to its enzyme by forming an amide with the amino group of a lysine side chain. The disulfide linkage of lipoate is cleaved when it undergoes nucleophilic attack by the carbon ion. In the next step, the TPP carbanion is eliminated from the tetrahedral intermediate. Coenzyme A SH reacts with the thioester in a trans-thioesterification reaction. One thioester is converted into another, substituting coenzyme A for dihydrolipoate. At this point, the final reaction product acetyl coenzyme A has been formed. However, before another catalytic cycle can occur, dihydrolipoate must be oxidized back to lipoate. This is done by the third enzyme E3 and FAD requiring enzyme. Oxidation of dihydrolipoate by FAD forms enzyme bound FADH2. The vitamin precursor of coenzyme SH is pentothenate. We have seen the coenzyme CoASH is used in biological systems to activate carboxylic acids by converting them into thioesters, which are much more reactive than our carboxylic acids. At physiological pH 7.3, a carboxylic acid would be present in its negatively charged basic form, which could not be approached by a nucleophile.